Okay, am I audible? Yes, sir. Yeah, thank you.
Yes, I can hear you.
Yeah, welcome, sir.
Thank you.
Hello, everyone. Yes, Professor. Yeah, sir, you are audible also.
Okay, I'm sharing the screen.
There is.
Hello,
Yes, sir.
shall I speak or you're waiting for something?
We are going for the inaugural and some introductory part within 8 minutes. Later on the 9 onward, you can continue with the session.
Okay.
Sir, can you stop your screen sharing so I can continue with some inaugural formalities? Okay, hello everyone. Here is Professor Sagar Ande, Assistant Professor at Dr. Rajendra Gode Institute of Pharmacy. I here heartily welcome to all the participants to international webinar series organized by joint venture of Dr. Rajendra Gode Institute of Pharmacy and Dr. Rajendra Gode College of Pharmacy. These institutions are a part of the Indira Bauddhishya Shikshan Sanstha, Buldana, with the global vision and mission to explore the pharmacy field. We are get the inspiration for such activity from our young dynamic president of Indira Bauddhishya Shikshan Sanstha, Buldana, Honorable Shri Yogendraji Gode sir. Indira Bauddhishya Shikshan Sanstha, Buldana is associated for running more than 30 educational courses wise pharmacy, engineering, medical sciences, agriculture, and several other. I am also thankful to this event co-sponsors, that is Unique Pharmaceutical Laboratories Daman, for their continuous support for arranging the technical program for faculties and students. Also thankful to Shri Parmeshwar Bangsar, Vice President Works of Unique Pharmaceutical Laboratory, for their continuous support. I am also thankful to the ZIM Laboratory Limited Nagpur for their support and guidance in the field of pharmaceutical research projects, and also thanks to Shri Prakashji Sapkar sir, Director of ZIM Laboratories, for their guidance. I am also thankful to Sun Enterprises Limited, Rwanda, for their support regarding to the studies of the global pharmaceutical market, and also thanks to Shri Sanjay Kumar Singh sir for their continuous support. Most importantly, I am thankful on the behalf of our institution to the eminent speakers for this international webinar series who give their valuable time to these sessions. Dr. Prashant Sakarkar sir, who give us a continuous support right from the beginning for the organizing this webinar. Dr. Balwant Chauhan sir, Dr. Rahul Deshmukh sir, and Dr. Ravikiran Panakanti sir. It's an honor and privilege for us that we get such speakers who having more than 25-year experience in the field of pharmaceutical education, research, and administration. Here, I welcome you all. Now, without wasting much time, I would like to request Professor Prashant Ajmire sir to give a short introduction about the today's topic. Over to Prashant.
Hello.
Yeah, you're
Hello.
audible, sir.
Good morning to everyone and good evening to our today's speaker, Dr. Balwant Chauhan sir. Uh, Thanks, sir, Sagar sir, for giving me this opportunity to introduce today's topic. Uh,
Okay, wisely, sir may get disconnected. I will continue with the topic. Today's topic is regarding with the our pharmacogenomics.
Sagar sir.
Yes,
Hello.
yeah, you are now audible.
Yeah. It is my great pleasure to introduce about today's topic. It is our first session of this webinar, international webinar series. Dear all participants, we all know that today's topic is current perspective on the pharmacogenomics and its importance in the pharmacy education and practice. The field of this personalized medicine affords multiple opportunities in the pharmacist, and pharmacists have specific knowledge, skills, and abilities that make them uniquely suited to advance the use of personalized medicine as a clinical tool. This all we know that as the field of pharmacogenomics rapidly evolves, the role of pharmacists in the education, implementation, and research applications of pharmacogenomics is becoming increasingly recognized. So, this webinar aims to provide an overview and current perspective of pharmacogenomics in the contemporary clinical pharmacy practice and to discuss the future directions on the advancing pharmacogenomics education, applications, and research in the pharmacy practice. Definitely, Dr. Balwant Chauhan sir will ignite the minds of all the participants, all these young researchers present in this webinar, and I am very much sure that all will enjoy and get the knowledge on this new topic. So I would like to Dr. Balwant Chauhan sir to continue his today's topic.
Wait a minute, sir. Yeah. Uh, Now this is the time I request to our respected principal, sir, Dr. R.L. Bakal, sir, to join for the virtual inaugural and deliver welcome words for today's session. First, we are going to go in for the virtual inaugural. I request Dr. R.L. Bakal, sir, to please press the button for the inaugural. Okay, thank you, sir. Here is now I declare that today's session and ongoing webinar opening ceremony is commenced. Now I would like to request Dr. R.L. Bakal, sir, to give the welcome words for the today's session. Yes, sir, continue. You are audible, you are unmute, and your video is visible.
Very warm welcome for the international webinar series. And for today's seminar, we have Professor Balwant Singh Chauhan sir as the speaker. It's my great pleasure to become a part of this program. First of all, I welcome our today's guest speaker, Professor Balwant Singh Chauhan sir. I welcome you, sir, as a principal of this institute and on behalf of my management. So, good evening, sir. And
Good evening.
And good morning to all participants for today's seminar. This event is only possible due to support of our guest speaker, our all sponsors and our president, Honorable Yogendraji GODE sir, and lastly, all participants, because only due to their presence, this seminar is possible. I conclude with hope. Definitely, this seminar will be helpful for all in future. Thank you. Over to Professor ANDE sir.
Thank you, sir, for your words. Now, I would like to request Professor Nikita Singhal, madam, to introduce our today's eminent speaker, Dr. Balwant Chauhan, sir. Over to Nikita Singhal, ma'am.
Good morning to one and all, and good evening to sir. Myself, Professor Nikita Singhal. Thank you for everyone for being here this morning. Today we are pleased to welcome Dr. Respected Balwant Chauhan sir, a very special guest from USA. We are overwhelmed by his presence. I will be giving a short introduction of Dr. Balwant Chauhan sir. Dr. Balwant Chauhan sir is currently working as an associate professor in Department of Biopharmaceutical Sciences at School of Pharmacy, American University of Health Sciences, California, USA. Dr. Chauhan took his BSc, MSc and PhD from the same college from Faculty of Science, the Maharaja Sayajirao University of Baroda, that is in Baroda, Gujarat, India. He obtained his MD degree in 1998 from College of Medicine, Spartan Health Sciences University, Wexford, St. Lucia. His PhD work was in the field of reproductive endocrinology. Dr. Chauhan became faculty as an assistant lecturer in 1970 at Department of Zoology at the Faculty of Sciences, MS University of Baroda, that is in Baroda. And later he was promoted to reader as an associate professor in 1983. He left for USA in 1987 and obtained post-baccalaureate certificate in toxicology technology from USA. Dr. Chauhan has involved himself in biomedical research since last several years. His research interests were first, sequencing the HAP1 gene, a purinic or a pyrimidinic endonuclease gene, a DNA damage and a repair enzyme. Secondly, effect of prolactin and estrogen modulation on another DNA damage and repair gene, that is O6-methylguanine DNA methyltransferase in context to breast cancer. Apart from this, he was also involved in generation of transgenic mouse model for study of insulin resistance. For a short time, he had also worked as a laboratory manager and a laboratory director for a drug monitoring and environmental toxicological analytical laboratories in USA. Currently, Dr. Chauhan's primary interest is in teaching human anatomy, neuroanatomy, physiology, pathophysiology, immunology, medical microbiology, medical genetics, and pharmacogenomics. Pharmacogenomics is today's topic. Before joining School of Pharmacy at Roosevelt University, USA, he served in the College of Medicine doing research and taught to several medical students at University of Illinois at Chicago, University of Chicago, USA. His teaching experience spans totally for more than 25 years and still many more years counting. Dr. Chauhan's current research interests include, firstly, pharmacognosy, in which screening of plants for their biomedical properties, especially anti-obesity and wound healing properties. Secondly, generation of rodent melanoma model and benzathrone's dermal toxicological aspects. And thirdly, pharmacogenomics. In pharmacogenomics, drug and drug plant extract interactions are involved. Then Dr. Chauhan has co-authored several peer-reviewed publications and scientific abstracts or posters. Some of them received our International Merit Awards, which is a great achievement itself. He had given plenary and invited lectures at several institutes. I can say one thing for you, sir, that every new experience brings its own maturity and a greater clarity of vision. So today he will be virtually sharing with us current perspective about the need of pharmacogenomics and its importance in pharmacy education and practice. With that, I ask you to give your full attention to Dr. Balwant Chauhan, sir. Dr. Chauhan, sir, please, you can continue.
Okay, thank you,
Thank
Nikita
you.
Singhal. Now I request Dr. Balwant Chauhan sir to guide us with their presentation on the topic pharmacogenomics and later on with interactive session. Sir, I request to start your session. Before that, I am going to log this meeting. If somebody is remaining to get joined, they can also join with us with the YouTube channel. Link is already shared on your chat box.
Thank you, guys. First, before I start my PowerPoints and actual teaching, I would like to thank for giving me the opportunity. It's my pleasure to meet you on the websites. Hope someday I come to your college and we meet again in person. That's my wish. So instead of wasting the time, I would like to start my PowerPoints, and I will say thank you again, each and every participant. I hope that from my presentation, which is the introductory discussion, you will get something. So let me begin with this. Okay, so can you see my PowerPoints? Hello?
Yes, sir.
Okay. So today's title that you can see here is the impact of pharmacogenomics in personalized medicine. This is a very new topic, even in the United States, but it's not that new in comparison to any other countries that we can see. Probably I have visited in England and in Germany, and yet it is in infancy. A lot more has been developed as far as US is concerned. So I'll go to the second slide. You can see here the President Obama in 2015, when he was giving his State Union, he mentioned about the precision medicine. And he invited that I am going to allot certain funds so that we can progress in this particular science. Purpose of this particular topic is I have something like 50 minutes, so I'll try my best. I have several slides. When the time is over, I'll stop at that particular point, and I may not be able to discuss a lot of other slides, but that will be okay. So I would like to briefly talk about what do we mean by pharmacogenomics? What's its definition? The second thing is why we should have a knowledge of pharmacogenomics. What is it that we would like to learn, we would like to teach, and we would like to put it in practice? So I would like to bring certain points which will aim to this brief introduction. The health provider equate the pharmacogenomics as personalized medicine. So some people understand this far better. When I talk about pharmacogenomics, those who have some background, scientific background, they will understand. But a common person, when I say the same subject as personalized medicine, probably it is going to make more sense. So both the words that you can see here, they are coined and they are very popular. The key to personalized medicine is pharmacogenomics. Why we say that pharmacogenomics is very important as far as personalized medicine is concerned? Because it has two elements. One, pharmacogenomics is on one hand going to describe various drugs. I will call that science as a pharmacology. So a base should be, people should understand various drugs, their mechanisms and all that kind. The second aspect that connects the pharmacogenomics is that some knowledge about the human genetics, or genetics in particular, that is a generalized term, but to understand what happens in human, a little bit more about the human genetics is required. So when we combine these two aspects, a knowledge of human genetics and the drug metabolism, their actions, why they are given and all that, when you blend together a new subject that is coming, we call that as pharmacogenomics. Pharmacogenomics is mainly based on genetic variability. And that's why, as I said, knowledge, at least a basic knowledge about genetics should be there to understand the meaning of pharmacogenomics, to practice, to do research, or to put practice in actual cases. It is very important. Then you will see the genetic variability. It tells that there is a variability. Variability means there is a variation. Where is that variation? When we talk about the genetic makeup of a human body or a human as a race. We have, let's say, 100 human beings. Genetically speaking, they are not similar. Their genotype will be different. Certain common genotype will be there. However, in greater detail, if you analyze genotype, you will see variations are there. That means every human is not similar as far as their genetic makeup is concerned. Within a family, let's say if I have a family and the family has 2 sisters and 2 brothers and all that, normally we'll say that they have inherited common genes. That is true. However, a brother's own gene will be different than the brother. The brother will have his own genetic pattern that will be different from the sister, and all 4 will be different if you see the detail of this variability. Because the variability is there, because they are different in terms of genetic makeup, their body is not going to behave similarly when certain drug is given. Sometimes certain common features are there, and that's why the genetic makeup, though it is different, the outcome of the treatment will be similar, because the genes which are metabolizing drug, they are not affected, though this variability is present. All the variability, all the part that I'm going to talk about, which are making variability, only certain they are going to affect certain drugs. So it is not required that each and every person of a family will behave differently when you treat them with a common drug. Then as I said in the earlier part, that genetics is the important part of this particular science. So simultaneously, if you see the history, World Human Genome Project was taken. When President Clinton was there, he realized, and he said that we are going to spend a lot of money, millions of money. And then the project which was given was known as a World Human Genome. Let us study the human genome in general. Those days, the technology was not developed. So they said that to decipher the human genome, it will take 3 years. Several scientists will work, but the project will be completed in 3 years. That was the initial catch. Then in Texas, one private company came up, and then they say that we are going to do this in less than probably 1 year. And when that news came, President Bill Clinton probably realized, or the advisor told this, sir, we have to have some kind of rules. Otherwise, this particular company will finally own the human genome. Human genome will be used in future by each and every country and all that. And then they are going to make money. So we have to stop that. So then Bill Clinton went to the Senate and he said, we have to have certain rules. If anybody who gets the knowledge of genome, it doesn't become their property. And because it is not their property, if any company who is going to decipher the human genome, US government is going to control them, that how you are going to put the price, who will have that particular knowledge, where you are going to put your knowledge, who can access, whether free that knowledge will be available and all that. So it's political aspect were also simultaneously worked out. And that gives an example, a country like India, that government has to control certain things and then see what is good for the humankind. And that's what the government's job is. And in this particular World Human Genome, that was done by the US Senate and the President Clinton. Now, there are certain predisposing factors, diseases that you see. So when you study the human genetics, you know, at least a biological part, biology, common biology, when you study Mendelian genetics and all that, we know certain diseases are there and their cause is the bad genes. So when bad genes are there, they are variation. I have that gene, my brother doesn't have a gene, we are not similar genetically. So it's a variation. The variation that you talk about, some of the variations are going to give the disease. Diseases are because of them. Some variations are there and they are doing nothing. They are just mutations, but if you see whether they create disease, answer is no. Whether any kind of metabolic pattern is established by them, then we can say no, it is not. So variations that we see here, we cannot just say all variations, all mutations are causing disease. Some variations are there, they are linked to the metabolism of drugs. Some variations are no way related to metabolic aspect of the drug. Some variations are there, they are not causing any disease. So when we see the variations, we have to identify whether this particular variation that we find it is related to disease, whether it is related to metabolic aspect of the drugs or not. And today's topic, we are not interested in cause of the disease. So the variations which are related to drug metabolism, is our today's focus. So first part, always remember, we have to study the variation. Second part, whether that variation is affecting drug metabolism or not. It has to be linked. Then only that variation we can follow and study, and then predict certain things. Specific genetic traits, you might have studied in Mendelian genetics that certain genes are there, certain alleles are there, they decide certain characters. These characters are known as phenotypes. So genotype describes the phenotype. In case of human, let's a gene for a skin color. Skin color is a trait. It's not a character, scientifically speaking. Within traits, variabilities are there. So my skin color has one gene, but the variability in the gene, my skin will be white, my skin will be brown, my skin will be black. So you will see certain traits are there, and they are nothing but linked to the genes, and variability in the genes, variability in the trait is going to relate to the phenotype. In this particular topic, the variation which is related to the drug metabolism, that's what I'm going to come at a later stage, how we are going to study and how we are going to relate, that I'm going to explain. So you will see which may explain many adverse reactions. So if we know that this variation is there, this is how it is going to metabolize a particular drug in that particular person who has that variation. Maybe because of that variation, adverse reactions are found with that person, and you treat the brother with the same kind of the dose, same drug for the same kind of diseases, the brother will have no adverse reaction. Then you are asking, I'm following whatever doctor is saying, it's the same drug, same dose, same kind of a disease, both the brothers are similar, but one has an adverse reaction, the other doesn't. When you find that adverse reactions are occurring, it's a concern of pharmacists, it's a concern of medical science, it's a concern of a doctor. Doctor will like to do that, why I'm prescribing this drug in a very right way, but this person has an adverse reaction. As the science moved, they related the adverse reactions could be because of the genes. These genes are metabolizing the drug in that person differently than the person or a brother where you don't see the adverse reactions. And the goal of pharmacogenomics is to optimize drug efficacy. So that is where we would like to have the importance. That's why we would like to have this knowledge. That's why we would like to see that our students get this, because they are going to put it in the practice. Limit drug toxicity is the second goal. So when we find toxicity, can we resolve that issue connecting to the variability and how our steps should be taken so that we can reduce the toxicity. So pharmacogenomics can be used to limit drug toxicity. That's why this knowledge is very important. To reduce overall cost. So our treatment that you are going to see here, and then you will say that, okay, how this financial aspect is involved. A simple way of thinking that a person has, and right drug is given, the person shows adverse reaction, and if you don't use, if you do not focus on pharmacogenomics as a physician, what's your option? You say, can I have any other drug? Okay, I'm going to reduce the dose, or I'm going to change the drug and all that. When you do this, you are wasting time, you're wasting human power, you are wasting different drugs, the treatment and all that. So when at the end, when you answer, before that you invested time, you have wasted a lot of money. If you think about the adverse reaction because of the genes and you can relate, then in a short time, you know, this is the answer, this is the drug that I have to give in this person, and it is going to remove toxicity, is going to bring the right outcome. So time is saved and the cost is saved. Fourth, why we have to have pharmacogenomics? Improve the quality of care. When toxicity is generated, it's not a good care. Nobody likes the adverse reactions and all that kind. So if we can shorten that, if we can overcome with this, then the quality of care is going to improve. So 4 targets are there, and that's why this science is very important. Then what has happened? What is happening is that if you go and study the history, when you ask different countries, those who are knowledgeable, pharmacists, doctors and all that we have some basic concept, but what's happening? And my last sentence is lack of appropriate education. We are not practicing this science because there is no knowledge. There's no education. Doctors are not aware of this particular science. Pharmacists are not knowing that how pharmacogenomics should be used and all that. So there is a vacuum because there is no education. Education is going to deliver the knowledge, and in that knowledge, experts will be generated who have knowledge about pharmacogenomics so that they can apply. So that is a prime thing. Just ask college in vicinity of Amravati and visit the pharmacy schools and ask how many experts you have in this particular science. The question will come probably only few or maybe none. Why? Because the part which is known as education, they got the degree, but in their education, pharmacogenomics was not there. That's why it's not that they are not capable to gain this particular knowledge or it is so difficult. That's not the case. Case is opportunity is not there. There is nobody education institute wise who is talking about, let me take this subject and as a course subject or an elective, teach this subject. So we have to have experts, they have knowledge, and then gradually you put that in a curriculum so that your students are going to know about pharmacogenomics. Then, as I said here, pharmacogenomics can improve the therapeutic outcome. That is the final word that I'm talking about. That's why this science is important. That's why I would like to teach this science. All pharmacists should know this particular aspect. Believe me, in United States, the survey has been taken, and the current survey says that pharmacists have more knowledge on pharmacogenomics than the physicians. The reason is that when I was studying my MD, my professor has no idea what is pharmacogenomics. As a medical student, I never studied pharmacogenomics. The lead was taken by the pharmacist, the pharmacy college. And that's why if you see this particular survey, they're worried. And that's why now the medical schools are also talking about very much the pattern that is adopted by pharmacy school, that in medical school also we should teach pharmacogenomics because doctors are also involved. It's not only the pharmacist who is involved in this particular science. So historically, lack of education is the reason, and many surveys that I have gone through, I agree with this particular sentence. And to summarize, medicine is what? Medicine is personal because individuals are different, their variation. That's why we call personal. We are all different. Some of our differences translate into how we react to the drugs. And sometimes it may not, depends upon what kind of makeup you have. This is why personalized medicine is important to each and every one. Those who are health related professionals, I'm not talking anybody who is teaching English or history, it is important. So when I say it is all or it is important, certain category of the health science workers. Why does someone need twice the standard dose to be effective? Doctors are treating and then sometimes the dose is increased and outcome will be there. Then the question comes to a doctor. Why does someone need twice the standard dose? Why I have to increase it? Then why does this drug work for you but not for me? It's the same drug, same chemical molecule. Why two people have a different outcome? Why do I have side effects and another person don't have? These are the questions. They appear in real life. And then a thought process was there. That was the birth of the personalized medicine. So these are the bullets. They are the questions, and there were no answers. And then slowly they started deciphering these questions. Why do some people get cancer and others don't? Getting cancer that you will see here has less to do with the pharmacogenomics, but I told you, if a variation is there, variation could be related to disease, and one disease that is nothing but a cancer. There are several that we can talk about. Why is antidoctoral information irrelevant to your own health and treatment? So this particular slide is nothing but curiosity, questions, and when they were answered, slowly a new science was developing. That science now we have given a name, is a pharmacogenomics. So let's see this particular slide. You have a particular number of patients that you are treating as a doctor, or let's say a pharmacy. A cohort is given here, all patients with the same diagnosis. So disease is same. Let's assume they have same sex, assume that same weight and all that kind. And because of that, a generalization, I'm going to use this drug, this will be the amount, this will be the dose, and I'm going to treat all the patients. When a doctor starts that, not all respond to the therapy. That is the reality. So what has happened? Majority probably you will see here that conventional dose is given outcome which is expected is there, no problem. But few from this, you are going to see severe overdosing, and then they will show you toxicity. Then you ask the question, well, I'm doing the same thing, which as a doctor I have to do it, however, outcome is different. So that's where the question starts. And then when a question starts in the science, you develop a hypothesis, you start working to find out the answer. You might have a case from this cohort, subtherapeutic levels, then expected outcome is not there. Maybe you have 50% outcome, but the 50% of the treatment is not working. You ask the same question. I'm treating the same disease, same cohort is there. Why I have this kind of outcome? Because this is the reality. That question was asked and science progressed. So at the end, you will see we should have a right dose, because you see one dose is not going to work. So right drug, maybe the drug which is given, and then you don't have anything that can help in the sense of pharmacogenomics. Pharmacogenomics doesn't have an answer. You have no choice. You are going to try another drug. Right indication, whether am I missing something? Right patient. Patient means the makeup, whether what I'm treating, the patient is right for my treatment, or patient is slightly different. And the time is also important, whether it is right time. So the last one, time, today I will not describe. Precision medicine is the future of medicine, that everybody believes that. The concepts are not new, because now 10 or at least 15 years are gone and people have browsed on this. That's why we say the concept is not new. But then why the progress is not there? Because the tools were not available. I gave you a project at the time of Blinken. I said that the new company came and they developed new instrument, they developed new procedure that was easy, time saving, and then the analysis were done faster. So you will see tools are important in science. A good example, let's say biology. Sometime you see the certain things with naked eye, you describe. Eye has a limit. Beyond that, you cannot see the detail. Some curious person said, I'm going to use a lens. Magnification, more details are there. Now you describe the same thing in a different way. Then they say, can I increase magnification? Microscope came. So see here, tools have to develop parallelly. If we don't have the right tool, we are stuck. Science is not going to progress. That's what it happened when a pharmacogenomics, we knew something like that, but to analyze that problem, tools were not there, methods were not there. And because of that, though the concept was developed, it was not tested. Then the tools are now developing. When tools are developing, companies compete, and then you will see when companies compete, more better tools, more cheaper tools, time-saving tools are going to come, and that's what is going on everywhere in the world. Then I have given this particular part just to see that if you are going to take this percentage of courses that recently adapted, so you are going to see this particular part is a body. This APHMG that you are going to see here is a body. And this particular body's function is, it is an international body. Then what is required, which is related to the human genetics. What can we do and how we can incorporate genetic various aspects into the curricula? How am I going to develop a curricula? And if you see here, the progress, you are going to see on the x-axis on the number 1, you are going to see the percentage, much more use of genetics now, if you consider, is taken by personalized medicine. So I don't know whether you can see my arrow, but see here. So if you think that genetics is 100 points, currently speaking, more points, more knowledge of the genetics is used by personalized medicine. The next one that you are going to see here, direct to the consumer genetic testing. Whatever the genetics knowledge is there, now how can I test this thing? It is growing, it's going faster, but you will see the first bar is taken by personalized medicine. On the other side, if you come to this particular aspect, you are going to see here the genetic counseling. Yet genetic counseling is not becoming popular. Genetics is there, maybe 0.1% of the genetics knowledge is used to develop this. How many college teaches you genetic counseling? Just see here. Even in a medical college, nobody learns that unless you are specialized in that particular branch. And as a doctor, you have to select, I need this higher specialty. Then you are expert. All the doctors cannot have a genetic. So it is slow growing. So you can see here, this current world is of pharmacogenomics. This is again, I would like to move faster. Pharmacogenomics covered in the medical school. This is a survey done by United States, and it's an old survey. You will see this was done, reported in 2010. Pharmacy school, 89% provided instruction in pharmacogenomics. Yet it's good, 2010. Number of hours provided overall in the pharmacy school, 40%, less than 10 hours. The other schools have 42%, 10 to 13 hours, depending upon the dean, the pharmacy faculty, how much weightage you would like to give, how much hours I would like to teach, and all that kind of stuff. And medical school, you will see 82. There's a big difference. Pharmacy school is leading than the medical. Maybe the medical school, the way it is going, it will be equal for some time. And if pharmacy people think that this is not my field, then probably medical school will take over and they will lead the whole science. Let's talk about the definitions and all that kind. So I'll just put it definition. There are two words. Be with me. Pharmacogenetics versus pharmacogenomics. The difference is here. One is genomics, one is genetics. So the most popular definition I have given here, that pharmacogenetics refers to the study of a gene. You are taking one single gene and then relate and study a single drug. When you do that, your subject is pharmacogenetics. But if you are studying all the genes, so many genes which are present in the human body, which is a part of our genome, and then you take the same drug, but not single gene, but now you are taking several genes, and then you say how they are going to affect a drug, that subject is called pharmacogenomics. Pharmacogenomics is more important than pharmacogenetics. Pharmacogenetics is these days used in research, but in practice, pharmacogenomics is used because single gene you can study, but other genes, if you ignore, your science is not perfect. These two slides are the other definitions which are given by Vogel gave a different kind of, Flaxis Fruel gave the different, but the most popular that I have described here. So I'm going to take this history because various scientists have their own concept and they describe. The most popular one, I gave you the idea. Little bit about the historical part. Gregor Mendel, everybody knows father of the genetics. That's why you cannot separate him, Charles Darwin and that. However, Motulsky in 1957, association of number of human genetic conditions with toxic response to drug resulting from mutations in the drug metabolism enzyme. So concept was developed. It was not tested the way it is in 1957. Vogel was the first who coined the term pharmacogenetics, not genomics, in 1959. Then Carlo, 1962. That's the time I was in India in 11th grade. That is the time when pharmacogenetics, a book was written. The world was quite ahead than what I was learning in India as a biology student. Vassal and Page in mid-60s to 1980s, genetic and environmental factors were related to this science. Large inter-individual variations were studied, and then they were related to metabolism and elimination. Science is gradually progressing. You can say 1980, I will call that the modern time. It's not an old time. And then people started studying cytochrome P450 system. They have to have some target for study, so which is more related to the drug. Being a pharmacist, you know, that's why they focused on that. Pharmacogenetics, the personalized medicines and all that. When you talk about pharmacogenomics or pharmacogenetics, I'm coming to the explanation of this particular subject. So pay attention. Now I'm going to talk about what is pharmacogenomics. To study that, you will have to have biomarkers. So if I'm a student or I'm a researcher or I'm a pharmacist or I'm a medical person, I have to understand what do we mean by biomarkers. Biomarkers are nothing but patient-specific genomic markers. And these markers that you will see here, and I'll just go briefly, we have several slides, so I'll just put it here, hoping that you have some basic knowledge. SNP, which is abbreviated and full form, single nucleotide polymorphism. one base pair has mutated, that gives you SNP. SNPs are the most popular biomarkers. So when I'm comparing one person from the another person, my question comes, what am I going to study first? The first thing is I'm saying I'm going to study biomarkers. Anybody has studied, I go and do research. If not, I have to make my own mind. I'm going to compare the genes of two persons and I'm going to see where are SNPs. Whether these SNPs I can use with a drug metabolism. If it is going to be connected to that mutation, I'm going to say this SNP is a biomarker. The biomarker is for this particular disease, this particular drug. So I have to have knowledge of biomarkers. And jumping on that, you can have a similar word. Biomarker is nothing but it's a molecular marker, because molecular science, the techniques are used to study these markers, not a biochemical assay. It's not going to give you that. That's why these are molecular markers, and the popular word is biomarkers that you will see. What is it? If you take the DNA as such, it is a string-like structures that you will see on a particular segment. Then at certain locations, these mutations are there. Let's say this is a single base pair mutation. I'll call biomarker 1, biomarker 2, biomarker. So I take a segment of DNA and I'm studying where there is a mutation. So I have one person, let's say brother A, the DNA, brother B is DNA. I'm having a complete genome, and now I'm going to say at the location 50 in this brother, G is there, in this brother I expect gene, both are identical, but if it's not, and if I have C, there is a variation, a single point variation. You can say here is a difference. Now, this difference is related to the drug metabolism. I have to carry out certain other structures that you will see here. So molecular biology tools you have to know. And the primary one that you should study is a PCR. That's why if you are a lecturer or an associate professor, assistant professor who is going to teach, you have to know some molecular biology. Teach your students what is PCR, what is the technique. The reason is you need a tons of material that you can take it to the machine. If you have a little, then you have a very much less. Probably you can do one testing, but it is not. So that's why if I have a DNA, I have to have so many multiple copies, because I'm going to use a certain amount of DNA. And because of that, I have to have a PCR. What is the second connection to PCR? That PCR, I can create primers and study the what we call it base pair, the sequence. PCR is also used to study the sequence. PCR also used to quantify your DNA and all that kind. That's why it's a basic. To see this product, you have to use electrophoresis. So in a basic teaching unit, you should have at least one PCR machine. You should have electrophoretic things, whether you want to run DNA, whether you want to do protein, southern blotting, northern, and all that kind. And then you should have array techniques, which is costly, and a trained person can only do it. The old way, if you are going to do it 1 month and you have one particular result, it's not going to make any sense. So I'll come that how that particular part was resolved by the industry and the tools were developed. You should have a concept of alleles. I told you trait depends upon a gene. I have a trait, let's say a skin color. And for that trait, I have gene. But all genes, if you compare that is related to skin color, they are not similar. There is a variability. This variability that you see in a gene that gives me a black color, that particular structure gives me a white color or a wheat color, whatever you want to call a brown color. When you take that, you call that the allele. Allele decides your phenotype. Allele decides what kind of a color of a skin will be there. Teach your student the definition of allele, give examples, tell them how you can identify. So now I have a knowledge, but then you have to have a practical. How am I going to analyze the alleles? Okay, I understand. Allele decides. How am I going to study? A teacher has to have that knowledge so that you can impart that. And then because these alleles are not similar, that's why different skin colors are there. So there is a kind of a polymorphic character. Because of that, your phenotype is different. And then you have more than one copy of allele. That's why we call that allele can have a polymorphic character. Because of that, it is expressed in a different way. Then that is varying from individual to individual. That's why in a family, same mother, same father, but you have 5 kids color will be not similar. It will be different, though the genes are inherited from father and a mother, because the same alleles are not inherited. Alleles, when you see in one brother, this and this color will be darker. The allele which is coming from father and mother will be different. Then you will have this color. So when we see in a pedigree, when you see alleles are not inherited. If alleles are inherited in 2 brothers of the same parents, they will have identical skin color, because allele decides this character. That's why we have to have a knowledge of allele, because allele is related to the outcome. The outcome that I'm interested here is the drug metabolism, that how they are going to modify my drug. And then if I jump to this, that, where can I search this? This is free. Indian people from India, you can ask it. So let's say if I want to see biomarkers, if you say, Dr. Chauhan, where can I see the different biomarkers? I say, go to the FDA. I have given here. You can see that it's freely available. And that's why this goes to the government. Government says that this cannot be always a business. And 2 ways these charts are created that gives you the valid biomarkers. One chart is called first adaptation, and I'll show you a short period. It's a long list. I've taken only few parts. And the second one is known as second adaptation. So what I'm telling, if I need a knowledge of valid biomarkers and study that, which drug and all that, go to FDA and get the first adaptation, a table will come. Or you can say adaptation 2, a table will come. I'll explain what is table 1 and what is this. I have given you here a particular link, that if you have this link, if you can copy, you do this, and then the table will come. So let's see here the table. So I'm taking here first adaptation table. This is giving me a biomarker, because science has moved. So I'm not going to find my own marker. Otherwise, I'm a researcher. That's a different interest. But as a pharmacist, so much is available to you. So your student will have not to. So see the first adaptation. First adaptation, the listing is done based on pharmacokinetics and pharmacodynamics. Your student, they know what are the 2 differences and all that. So see the first column. It says the pharmacodynamics. What's the biomarker? ALK, which is the drug. The name of the drug is given. And if I say, what is this marker? Then I have to go and find out a genome. I'm going to find out the marker. The marker will give me, here is a small piece of a DNA. And if you find that, this is how this particular drug is going to behave or is not going to behave. The drugs category is given oncology, and then package label. So if you open a package label, will give you this indication. I don't know currently that in India whether the package label is going to give you this detail or not. But then science advances. I hope one day Indian people, the pharmaceutical company, will also include. Let's see what is the table A2. As I said, both have similar things, but the way you attack the table is different. So this is therapeutic area. So I said, okay, I would like to see the biomarkers and all that, but I'm confused. I don't know about the drug and all that. So then I can say, well, what you would like to enjoy? The person will say therapeutic area, analgesic. Dr. Chauhan, tell me the biomarkers of analgesic. Then take the table. That's called A2, because the listing is done by a therapeutic area. You are working on painkiller, you are working on a pain, you are working on something else. So you go and identify, and then you come back. Both the tables are overlapping. At one point, you can connect the table A1 to A2, but how you would like to search? So I have given you this, that it is freely available. Let's come to the genetic variations. How many different kind of genetic variations are there? One variation SNP, which is single nucleotide polymorphism, which I said here. Another variation is cis transregulatory variations. Another is insertion or deletion, or both together, indels. The word indel, you can see here, from insertion, IN is taken. From deletion, del is taken. So when you see indel, it has a deletion and it has an insertion. But in sometimes the DNA molecule only have insertion, or only some part is missed, only deletion. Some DNA will have both. These are the variations. So one more time. What is a variant? Variant means I ask the question. Two brothers, they are not behaving similar way to that particular drug. Why? Because their genes are different. Where is that difference in the gene? The DNA. I have to find out that difference. How can I find out? Well, study SNP, cis transregulatory regions, study insertions, study deletions, study indels, or study copy numbers, and haplotypes and tag SNPs. When you have a haplotype, it's a greater detail. At the end of a haplotype, I hope that you know this particular word. Sequences on the DNA will be there, and a small piece will be there. Haplotype, haplotype, haplotype. You might have studied in your immunology class, or blood grouping. The blood grouping is there when you transfuse the blood and all that. They are haplotypes. So when you see that, and then they found out that if I have haplotype A, haplotype D, haplotype T, and at the end you will see S and P, and they say that if I have 3, S and P is similar. Sometimes it is similar, sometimes it is not. So if I know this SNP, I can make a prediction. The haplotype will be like this. So that's why I tag that SNP to the haplotype. But to tag SNP, first you have to study haplotype. So this is where the differences will be there when you compare the genome of one person to the second person. For each one, you have to have molecular biology knowledge, you have to have some background in genetics, and the third thing is, what are the methods? How am I going to study? When you complete that, you have prepared your base. Then copy number variation can be detected with microarray technology. So I'm not going to describe each and every technology. I'm just going to give you one and see how the science has moved. So let's say I want to study copy number variation. My doubt is that copy number variation is related to drug metabolism. I have two brothers, and this brother has certain copy number variation, and then probably that is interfering with the metabolism. How am I going to study CNV? Well, I'm going to go to the microarray technology. And the microarray technology, which was developed, they were basically developed for CYP, cytochrome P450, UGTP, uridine glucurosyl transferase, GST, glutathione S transferase, and SALT, sulfur transferases. But today, there are tons. This was the beginning, and which were more popularly related to metabolism. That's why science started with this. But I'm not telling you these are only 4 that you can see in a microarray. Microarray, 1,000 and 1,000 different types are available. So let's see here that what is that you will see. Other methods are there to study that. And one is called pyrosequencing, another is called multiplex PCR analysis, TechMan analysis. I'll show you something about TechMan analysis, lab chip microfluidity technology, and technology has increased and increased. I've taken the most popular one, but I can put 20 different technology that you can use with the pharmacogenomics. And the best technology that you should see, how sensitive it is. So when I compare one DNA with the second DNA, if one base is changed, single amongst thousands, and if your technology finds that change, your method is best. Your method cannot find that, but let's say my method is there where I can detect 10 or more. Yet it is good, but it's not perfect because 9 will be missed. Your technology is not sufficient. Today, most of the technology which are used can detect even a single base pair. Imagine your genome length and the size, even at one point, is going to tell you. And statistically, it is believable. It is that accurate method, and it is all automatic. Then you are going to see, I'm just going to tell you what is microarray technology, the Roche molecular diagnostic, which produces so many things. They came up with the chips, and the first chip, this was the enzyme they studied, and their genes were plotted. Second one, which is the most popular. So if somebody says as a teacher, Dr. Chauhan, where should I start? Which enzyme that I should select to teach? I will say CYP2D6 and CYP2C9. These are the most common. I have taken out that particular slide. But if I put all this P54, which are the ones which are so popular and which have been mutated, these are the two giants. Others are involved, 2%, 5%, 20%. These are more than 70%. That's why we should study them and all that. And the method has to be approved by US Food and Drug Administration when you are using it for any purpose. For research, you have not to. Then in this stage, PCR amplicons are generated. So I'm just describing what is this Roche model that you'll see here. Then it is fragmented. Then you label it, hybridize to a microarray, and bound products are stained. And then you have a machine, you take your slide to the machine and it will automatically read. And then whatever the genes that you are studying, the variations, you call AmpliChip CY450. If I'm interested in CY450, I have a choice to buy the AmpliChips. And I'm telling you, a custom made can be done. And there are thousands or more than 1,000 AmpliChips are available these days. And you can see here the advantage of this platform, that many allelic variations are tested side by side. So I have not to take one variation and do the whole process, I'm done. Start from the scratch, do the second. No, the machine at the same time. And I'm telling you, 1,000, 10,000 at the same time, it will give you the result. It's so quick. Economically, it is far better. So that is the reason why we have to have an AmpliChip and the array rather than doing the small basic molecular science. So I'm just, you can see here, association between 500,000 or more SNPs and a specific disease and a drug in quick, less time. Machine will give you this detail. And it looks like that. This is only a simple, and there are different kind. So I've taken only Roche. If you have not seen the chip, how it looks, then you have the prepared RNA. So you have to take the RNA for the variations or experimental conditions, 2 different cancers, whatever your normal sample is there, normal RNA. You have cancer, you have taken tumor, and then you have taken your RNA, and then you are going to label what I gave you the process, and then you come to the chips, and then you use it, you go to the machine, and then a point mutation graph will be there. This is a colorful graph, and then if you expand, the result will look like that. If I have a microscope, and if I see that the chip, that particular dot, and expand in a big way, is going to look like that. And what are these colors? Colors are interpreted. Chips are of different variety. You can see here, in this case, several things are there. I have obesity gene here. I have a diabetic gene here. It depends. You ask the company, and they are going to give you the chip that you like. And in a magnified view, normally with eye you don't see, but the result is that each one that you see here is your probe. That's your DNA fragment. And what this color indicate, because you have fluorescent dye that is used for that. So instead of going this, but let's come, so I can just tell you what these colors are. So let's say you have a normal cell. It will be a green color. That means your DNA has not been mutated. It's a normal thing. So if I see this, if let's say this is my DNA, oh, I'm a normal. This is your DNA, you're normal. This is Dr. Bakal's DNA, it's normal. See? Now you have Nikita, and I see this is Nikita's sample, and it's red. It says in pathological cells only. She has probably the cancer gene. This particular dot tells me which gene it is. And I told you that in one chip, you can put 10,000 different copies on the slide at one shot. So if I say this cancer will have this variation, this variation, this variation, this variation, in one shot, I will know which variation is there with Nikita. And it's all computerized. Your data is given to you and you analyze. I don't have time to go in a detail. I have a few other things to say. So let's say the second part, genotyping by TACMan chemistry. The TACMan word that came because of one computer game. The main word that you will see here was a computer game. The theory behind, the hypothesis behind operating that game is similar to this. That's why they named TAC. TAC is a enzyme's name, but the technique is TACMan. And then in that case, you process the whole things and all that. And then you have, let's see, CYP2C19 is your enzyme with the allele. I'll come back to this and all that. And you use several kind of a fluorophores. Every fluorophores that you use gives you a different color. And then you flow cytometrically, you study that particular part, and then you have used Vic as a fluorophore. And then when you see that, it tells me it's a wild type. When I have a FAM that you are going to see here, and again, it will be a different color. Marshall will take that and is going to put it. When you have FAM, you can see here that allele number two, two copies are there. So you know in genetics that one comes from father, one comes from mother, homozygous. Two genes, two alleles are there with that person. When I come to this, it's a wild type. That means only one copy of that gene is there. And that is in a large population. That's why I say that if I have that variant, you are a normal person. When I see that wild type and wild type that you are going to see here, then I have two copies. It's a perfectly normal. This can be like a career that you might have studied because one copy is there. Sometimes this one copy will give you some character. Sometimes it has no meaning at all. But this definitely you'll see that gene has to be related. And then the people who know about that, you have control, you have water, you don't expect any signal. So your technique is working and all that. So in short, this is the one where quencher is used, how the technique meant. So I'll just move it to the things. I have taken this up to 2000. You can see here, one analysis those days cost this. Just count the zero. That was 2000. In 12 years, the price to do the same analysis came to 10,000. If you will ask me, Dr. Chauhan, where we are, if I put it here, 2020, $200. Whole genome. You can use for any purpose. Today, you say, I want to use for cancer. 10 years, the patient comes to me, and then I see something different. I'm not to do any analysis. I am going to use the same data. I have, after 10 years, hypertension. I have the same data. It's a computerized version. I just manipulate with a software and then manipulate the genes. $200 is done. That much science has been advanced. So other part, I will just give you. Sometimes you will come across GWAS. I don't know how much time I have, 9:28. So GWAS is a abbreviated word, genome-wide association study. I told you that SNP is a one mutation, and I'll come and I'll explain the language. So if you have that, the question is whether this mutation has any kind of a relation. It could be a relation to the disease or no disease. It could be relation to the drug metabolism or no way. It's just a mutation, useless. I'm studying it, but I cannot relate. How shall I know? So when I have SNPs, let's say I have 2000 SNPs, my question is, which is actual biomarker? This is the one. This is the mutation that gives me metabolic disorder. All others are not. This one is related to disease, not to the metabolic condition. What I'm doing it here, I have SNPs, I'm trying to match. Is this matching with disease? Is this matching with this enzyme? Is this matching with this drug and all that? When I match it, it goes to genome-wide associated studies. This is a body. This body, I have that knowledge. I go there and I say, this is my knowledge. Please deposit there. Once I deposit there, it's available to each and every part of the world, completely free. In introduction, one professor told that Dr. Chauhan sequenced a gene, a epironic gene, and sequenced that in Portland. As soon as we sequenced the whole gene, we submitted our sequence to the bank. You go there, you don't need my permission. I sequenced the gene. The gene is like that. So that is different kind of this. For this is a mapping. And when you map, we are happy. If the mutation is not mapped, you have scientific knowledge, but it's useless because you don't know what that mutation is going to cause. And when this mapping is done, they create a plot. That plot is known as a Manhattan plot. And I'll show you that particular. And this particular plot has a drug which is clopidogrel and all that. I'll bypass that. I would like to show you that. So see here, these are chromosome number. I have DNA from chromosome 1. I have chromosome number 2 on the x-axis. Then all these SNPs which are related to the clopidogrel, that particular drug, platelet inhibitor and all that clotting part and all your pharmacology that you see here. Then as a scientist, I have studied. I said, okay, I saw these kind of, let's say these are 1,000 mutation. I say it is not related to this drug. Okay, chromosome 2. Yes, I have so many mutations. This one is related. Why? Because I have established a norm line. Anything goes above, I match. If you have this in this quantity, you will have this. This is your outcome. So Manhattan plot gives you which one markers or SNPs are mapped. So once I have done this, I'll go to the GWA and say, I have done this research. Here is an SNP related to this. Here is an SNP related to this. I have matched. These are all unmatched. I don't know what these mutations do. I have no clue. So Manhattan plot, how to analyze and what it tells should be known. This, I will just pass it on. Pharmacogenetics, I've taken certain questions that, let's say, what basic knowledge you should have. Somebody says, Dr. Chauhan, come here, become a director of pharmacogenomics. Then I'm going to say, I need this, I need this, I need this, I need this. If we don't have this, please don't waste your money. So this is the slide, a priori. Some knowledge has to be there that I can use. If I don't have a knowledge and I just say, let's have a building and this and that, you're wasting your time. It's linkage. I should know that whether for a human population, for the South Indian, Sardari or whatever you Maharashtrian, these and all that, I have to have this linkage before I practice. If I don't have a linkage, it's just a mutation. I'm going to say, oh, I found this change. And then you will say, how can we use it? I don't know. See, so that's the second one. The second is pharmacodynamics knowledge and relation that you have. So I have just listed, I would bypass because I have a few other interesting slides. But this slide tells that to practice pharmacogenomics, you ask me to become a leader. I'm going to say, I have to have this, I have to have this. If I don't have, it becomes my duty to take that knowledge and put it in that center. Then prediction, because of this, what can I predict? Utilization, whether it's costing. So I have pharmacoeconomics. I'm going to say, this is the test, this is the expenses and all that. And can I use that in AMRAVATI, that is a hospital. And the cost person case, sir, to do this one analysis, there will be 5 lakhs or maybe 1 crore. Useless. Who is going to give me that money? So I have to come at a later stage. Political considerations, ethnic considerations. I have to see the cost. If it doesn't match, it's just a knowledge. You cannot put it in a practice, period. So all aspects you have to have an expert with you so that you can take these people and then run your show. I'll pass this, let's say, concept of genetic polymorphism. So I'll just give you the definition. You might have studied a variation and all that, but actual definition. It is estimated that the genome of any 2 given individuals differ by approximately 1 nucleotide in every 1,000. So if I have a DNA this long, and if it is 1,000, I expect 1 mutation. Another 1,000, another mutation. There are possibility I have many, but generalization, 1,000 nucleotide pairs, 1, or a difference of approximately 3 million base pairs from one corner to the another, whole genome. Why I have put it there? See the complexity of the science. You have 3 million base pairs which are not matching between 2 brothers. How am I going to use this variation to make any sense? 3 million base pair differences. Which are useful, which are garbage and all that kind of stuff. It looks different because people have no knowledge. If you have knowledge, this is not a problem today. Science has moved so far. We are not afraid of millions, 30 millions or 40 millions. Then you will see the different ethnic groups and all that frequency of polymorphism. Normally it is there. So when we would like to study, then I'm going to select well, let me study ethnicity. Sindhi people, Maharashtrian people, these people, that people, Muslim people, Sardarji, Jain, and all that kind. You have to have something ethnic. And once that ethnic group is there, some commonization will be there. But if I'm going to study, my one patient is Sardarji, one patient is Maharashtrian, one person is Brahmin, I'm nowhere. See? So you have to have something as a vision how you are going to, in that population of Amravati or in that area, how I'm going to tackle that. I have to have this kind of the things so that I can serve at the end that particular community. Okay. When a variant is so common that it is found in more than 1% of chromosomes in general population, so large number, general population doesn't make 1,000. Okay, it's a biostatistic. The variant constitutes which is known as genetic polymorphism. This change that I have found, that change has to be 1%. If I have 0.8, 0.9, and if I have, let's say 5,000 people, then I say they are equal. There is no change. Absolutely, the genes are different, but I say they are equal. Don't bother about this. It's something like that. I have a brown color, and there is a little brown color. Basically, I'll say, oh, they are brown. You can't say both are different. Then I'll say brown, black, this and that. See here? So the definition that we accepted, that polymorphism that you see should count in a wild large population more than 1%. Then I'm going to focus. When I see the variation, when I do the statistic, if it's less than that, then I say all people are similar. Forget about it, because I don't call that as a variation. Then what I'm going to call those, then I'm going to say these are mutations. These are rare mutations, which keeps on occurring. In my life, if I analyze my DNA today, and after 20 years, the variation has changed. I'm exposed to radiation, I'm exposed to chemical things, doesn't mean I'm different. But my change, when I count with a population, all other people, and if it is more than 1%, I'm a different person. See, that's where my definition is. That's where I say, okay, I would like to study you. Otherwise, it is just a mutation, and you study as a mutation. You don't use that in pharmacogenomics, that particular. So genetic variation that you are going to see, and it can occur in anywhere. So let's say you have a DNA particular part. I'm talking about one gene. Upstream, your start codon is there, your intron, exon, and all that, your AND and UTP and all that. So I hope that you know one gene structure and all that. This genetic variation can be anywhere else. It should not be only in the genetic part. Variations that we study, which are related, which are important for drug metabolism, they can be anywhere. Remember that. They can be upstream, they can be here, they can be in exon, they can be in this, and there is no rule. If it is a part of your regulatory gene and all that, then it is more important because it will affect your gene. So your basic knowledge, where this mutation is there, you have some knowledge to put it to pieces together. Okay, probably it is here. Genetic part that you see here, variations are very rare. Always remember. The regions, so I have, let's say, I hope that you see, I have this finger part, this DNA, this DNA. Let's say the middle part is the actual gene. This is not gene. Most of the variation that we now know, they are not part of the gene. It is with the other parts where gene is not there. So you take that long stretch, there's no gene here, but then if you say, is this variation important? Answer is yes. Why? Why the mutations in the gene that we don't see? The reason is, if the gene keeps on mutating everywhere, I'm no more a human. Our genes should remain stable. If my life is for 70 years, if my genes keeps on changing, the gene which controls my metabolism and everything. That's why that particular part, nature has protected. I have insulin, it's a gene. If I keep on mutating, my life will be gone. Where will be the mutations? When the gene is not there. So when I attack, when I have a DNA, when I want to read, I'm not focusing on a gene. My major things will be non-genetic part of your whole genome, which is more than 90%. That's where I'm going to focus for these variations. Okay, so let me move to this. Okay, I'll just tell you, just for the SNP, I told you there are several markers and all that. So for example, the single nucleotide. So this is a base sequence, AGGTCA, and then you see GT. Then I have another DNA piece, and then I see that particular part. And now you see here, A is similar, G is similar, G is similar, T is similar, C is similar. But here is A and C. One nucleotide change, S and P. And then I have GT and I have GT. how you are going to write SNP is an accepted language. Then SNP, I'm going to write A. This is not more or less. This is how the notation. Then it is G. That tells me A was there, and then it has become G. Now, let's say you want to add, which is wild type and all that. So see here, sometimes you are going to see at the same location, not only one G, and then I will have something like this: A, G, G, T, C, A, G, T, A, this is G, and I have another one where I'm going to see A, G, G, T, C, and instead of G, I will have something else. So now I have three. When you have that, it gives you three allelic variation at the same location. I can have four variation at the same location. If this number is 2020, and if I'm studying, I will have A, I'll have G, I'll have T, I have this, and the allelic variations will go on. And when you do that particular part, the first instance they found out was the MDR1, which is multi-drug resistance gene. Actually, this is the gene that was sequenced first in our history. And then if you will see, the wild type that you will see is a G. So now in this case, G. So technically speaking, if I say now, if I ask you which is wild type, A or a G, you will say A. You write wild type first, and then your variations are there. What is wild type? Wild type means in a large population, if I have 1,000 people, most of the people will have A at that position. Only few will have, that means they are not wild. Sometimes wild will say they are normal people, because so many people in a population are there. So this is how you are going to. So when you have a triplet, then you say G, you write T, and then you write it. If I have another one, a slash, and I write another one. So you have to learn the language. So then you can understand the publications which are coming, and then you don't know, then you will read it, but at some point you need an expert. What is this which is given in there? So when you read pharmacogenomics, real articles, you have to have a good base of your genetics. Otherwise, half of the paper you are not going to understand. The another part that you should know is a RAF SNP, which is a reference number, freely available. It is a database that you are going to see here. It's a database and all that. So if you come to me and say, Dr. Chauhan, I have this and that, then I say, okay, let's see the reference number. So I'm going to the main library, and then it's a US government library, and then I go, it's a software that you use it. It's like a storage which is available to you. And then this is a site which is freely available. You can do it from India too. Then you have certain interest, you should know the reference SNP. This is the one I'm interested. All the details will come. And currently, how many databases are there? Just see here, 55 million SNP submissions are done. Out of that, 14 million are the references. The others have no clue. They did the research. I found this. Okay, it is here. I found this. It is just telling there's a change, there's a change, there's a change. But what is it? Nobody knows. So that's why you have to have RS number. So if you are doing research or something and you want to do this, then RF SNP's number, if you have there, then it is going to give you that. And this particular number I have given, 2008, is our old data. And then see here, if I'm interested in SNP in the MDR1 gene mentioned above, then cataloged is this. I go and put this number, I will have all the data coming in front of me. Then you have to use it, what you need, what you don't. But you are not to go and flip the pages, So SNP, you can see here, the variation is done here. The change has been done that I talked about, and I explained the nomenclature. Now, if I have to complete this, I told you 7 types of variations. You have to know each and every variations in their language, because SNP is one. I told you deletions are there, additions are there, and copy number variation. There are 5 different copy number variations are there. What are those? Whether you are interested in that or not, where that detail will be available. It will be a free detail will be available to me or not. Okay, nomenclature. I'm just giving you whether you are with me or not. A, B, C, B1, 3, 4, 3, 5, C, and this. So you can see here wild type it in most of the population I expected number 3, 4, 3, 5. So I'm counting the base pair 1, 2, 3, 4, 5. I'm here that part of the DNA 3, 4, 3, 5. Most of the people will have C, but there are possibility that variation will be there. It will be T. But what is this gene that I'm talking? It is ABCB1. It's a name of a gene that you can see here. Okay, first letter that you see here is always gene. Then location of the DNA, that is your number, the number between gene name and the nucleotide. So exactly at that position, you know what you expect. First nucleotide letter that you are going to see, original nucleotide, a wild type, is also known as a reference type. And then C is the one that is, if you find that, it's normal. It's in a large number of the population. Second nucleotide letter that you see is a J. That's your variant, SNP variant. Then you go to the GWAS studies, and then you say whether this makes sense or it is just a mutation. Will anybody say something about this mutation? Is it affecting disease? It is affecting cancer? It is affecting by a drug, which drug, and all that kind. And the story will go on. Okay, tell me here, like a student. Okay, I'm treating you like a student, so you bear with me. An example of SNP is VCO-RC1. 1137CT. Based on the pharmacogenomic nomenclature of this SNP, what is the gene interest? What's your answer? A, B, C, or D? Answer is B, because that's your gene. This is the number. This is SNP. Okay. Alleles, definition of alleles variant. This is a basic generics, so I would like to just move it, but you should know that we always have two alleles. Why? Because one comes from mother, one comes from father. Remember that, unless there is a deletion. Otherwise, every person will have two alleles. It could be identical allele. One thing is different, one thing is different. So within allele, you have a family, subfamilies. Within subfamily, you have sub-subfamily and all that kind. So that is a nomenclature, and that's the detail about the alleles. And when you teach to your student, this is a must to teach. Otherwise, they will not understand the whole subject. So this is nomenclature. Probably many of you, I hope so, 99%, your teacher or you might have studied. So cytochrome P450 CYP gene that you will see here, that I'm now taking here to teach you the nomenclature, the language, CYP2 C19. Then what is this particular part? The first part tells you is a superfamily. Okay. Then the family that you see is 2. This is the way you have to write. It's a language. This is accepted all over the world. You cannot create your own things and write and say, my paper should be published. They are immediately going to say, hell with this. I don't understand what the language you are using. This is the way you have to use it. Then the C1 is your subfamily. The number after that is your individual number. Then when you start writing about allele, you put asterisks. Anything after the asterisks is all allelic detail. So the 2 is an allele. Then A is a suballele. Suballele has a subfamily. Then I'm going to say A2, and it will keep going on. So why I'm talking to this? Because if you read some literature, you have to know this language so you understand that what these people are talking. Let's say example of 3 allele subtypes. So you can see CYP2 19, asterisk 1, 2, and 3. I have 3 different kind of alleles. That's what I'm looking at here. How the science labeled 1, 2, 3. When asterisk 1 is there, it is a wild type. Wild type, always that allele, you write 1. That's the rule. So in this case, this is a wild type. So if I have 10,000 people, most of the people will have this, and few will have 2, few will have 3. So the language that I have just talked about. Let's say I have a subtype within that, then I will say 1A. So I can write another one, CYP2 C19 asterisk 1A. I can write another one, but then it will be a number. I can write another one that will be alphabet. And within alleles, copies and copies and copies and differences, because you can imagine how much genome is there over the globe. No person looks alike. That much variation we are talking here. And that's why this becomes so complicated, because tons of variations are there. So let's say genotype that you are going to see. This tells me that 2 copies of the normal allele is there. Why 2 copies? Because one came from father, one came from mother. Let's say I have this that I read, 1, that tells me I have 1 normal copy with me, my genome, but another is not normal. It could have come from my mother, it could come from my father, or spontaneous mutation. And when we study this, now you match. Okay, 17 allele is there. Can I? So see here, metabolizing status that you are going to see here of the P452C19 gene, normal metabolizer. So if I have this, you give the drug, you expect outcome, everything will be fine. But if you treat me an extensive metabolizer, that means my combination is this. If I have this genetic makeup, then I'm going to metabolize that drug so fast and clear it out, I will have no effect of our drug. Then doctor will say, God, I'm giving the right thing. Why the patient is not responding? Because that person has 1 asterisk and then 17. Now this is matched. 17 extreme, that anybody who studied match through the GWAS. Initially, you don't know what is 17. You have to make studies, and then you have to prove it that if I find this allele, this is how it is going to express. If I have 2 copies, ultra rapid metabolize. In this case, you give the drug and clear out. If I have a normal 1 copy and any of this asterisk 2 or asterisk 3 or asterisk 4, so many are tried, and then all are representing intermediate metabolism. So it metabolizes the drug, but not that fast. So some effect will be there, but full effect will be not there. If you have, see here, no normal allele, you can see asterisk, there is no 1. All other things which are given here are abnormal. And your second copy is also abnormal. When you have, let's say, 0.2b and 0.2, you have 0.2b and you have 3. You have 3 and 2, you have 3 and 3. All combinations are there. That means you are poor metabolizer. You will have toxicity. You have a drug which is given and body is not metabolizing for a long period. It will remain and it will shoot as far as quantitation is concerned, and then toxicity will develop. So when that happens, then you say, let me study. You go and see the genetic map, and you know your patient is like that. You have outcome. What to do? That is the second part. Once I know what is to be done. So in a simple way, I'll explain. So let's say functional allele is asterisk 1, wild type. When I have 2 and 3, loss of function allele. Function is gone. Your enzyme is not working, period. Then you have this combination, increased activity of allele. It is going to metabolize your drug. So you have this combination. I try to give you how we make the decisions that you can see here. And then homozygous, heterozygous, that's your pure genetics. And then you have to study if 2 copies are there, what's going to happen. If 1 copy is there, what's going to happen and all that. Okay, so I'm going to move faster. Now you determine enzyme activity. The patient genotype is homozygous with a normal functional activity. So I think this is a test. I don't want to take the test now because I don't know it. So here is a question and the answer is given. I'm going to pass it. To generalize, let's say you have a cohort of these people that you are treating it and all that, and your drug which is given, let's say 100 mg is given to all, and that's a normal way to treat. That's how you are going to start, a doctor, a pharmacist and all that. And then your genotype, which is given here, and this is a small piece of DNA, and then you have this combination, or you have 2 copies. In this case, only 1 copy is there. You can see both the copies are there, or sometimes another one is there, 2 versus asterisk 3. You have asterisk 3 and 3. You have asterisk 3 and 4. Any combination is there. And your phenotype out, if you have this combination, ultra rapid metabolizer, okay, then you will see your drug which is given is cleared. For this, you increase the drug. Then doctor says, let me try 500 mg. Let's say you found out this extensive metabolizer because of this genetic pattern, and you have several which is going to give that. In between this, that will be your drug of choice. Doctor will decide, I should start with 300, 400 or this and all the kind. Intermediate metabolized, then less than 100. And if you're a poor metabolism where the toxicity, you cannot give 100, then you will say, let me give 10 mg and see what happens. So you used your genetic analysis, you use how the metabolism is there, and you decided how you will treat your patient and what will be your outcome. Okay, what is this? Okay, I'll pass that. And let me see. Okay, I have 5 minutes, and then they said I have to take question. So I'll wind up, but I have several slides, but this is important. So just bear with me, I'll take your question. You will ask me, Dr. CHAUHAN, in America, if I want to study all these things, where should I search the information? Then, this is very, very, very important slide. There is a site called Pharmgkb, gives you tons of knowledge, and I have each one. If I have time, I will at least describe one. You have CPIC, Clinical Pharmacogenetics Implementation Consortium. 5 centers will have been established by the government, and then they said, you do this, you do this, you do this, and they keep on doing research and then analyze, map, and then they give guidelines. One center in Columbus, psychiatry drug, that's what it is. You studied here only the cancer drug. And then information comes. If you want to see those information, you go to CPIC site. Then you have IGNITE, NIH, and GIRI, National Human Genome Research, which is for pharmacists, for researchers, anybody. It's a general research-based site. Then DPWG, it is available in Europe too. It's a combination with USA and Europe, specifically Germany has participated to generate. FDA labeling, I showed you. You can go to PGDK, St. Jude, children disorders that you see, then this is the one that they are doing research, and they have a lot of information given. CDS, Clinical Decision Support, then French National Network. So for other countries, I have not listed, but you have to go with those countries and find, and probably you have information. So what I'm telling you here, okay, I need this information. I'm telling you, please go to Pharmgkb. Maybe you have your answer. If not, let's go to CPIC and search that. So that is what I'm giving you. And these slides are telling, I have taken an example. If you go to Pharmgkb, what informations are there? Because I don't have a time, I don't want to go. But you understand what I'm talking, that every site is uniqueness. And what is this Pharmgkb? Genomics, they have G, knowledge base, gives you this. Pharmacogenomics knowledge base. Your all informations which are known to the world at this time in the United States are logged here. How to open up? Here it is, www.pharmgkb.org. You click it, and then you start reading, and then you will learn about that. It will give you a lot of information. Then another CP guideline. So because I'm running out of time, I'm going to flip it over. And if something is there, I'll just click, or otherwise I'll wind up. How the GKB page, if I take that, how it is going to look like, that is given here, because I don't have a time. But you can see here, GKB gives me a lot. Genetic test, which is a company, what genes that I can study. So if my interest is here, this information says go to this, and you have a chip. Only thing you have to know how to use, but information is here. Then it tells you that which drugs you are working. So let's say your patient, you have a problem with clopidogrel or whatever, phenytoin. Then you say, oh God, it's listed here. Information is there. Start reading, and it will tell you. And if everything is given, you have not to do anything. Read it, interpret, and take your patient and go with that knowledge. So these are the sites that I have listed here. You can see the second page of that. For this, you have to sit, you have to read and learn. You cannot just do it in 5 minutes. I'm just giving you highlight, overview, where the science is. You can see the dosing guidelines. So now you will say, okay, I know this variation and all that, but how can I decide the dose? Okay, go to Pharmgk, it will tell you. I go there, it will give me the dosing. I'll click here, boom, boom, and it will give me all. Read it, what is applicable, you read. This is the gene and all that. So you can see science has developed. It's not fully developed, but a lot of things are there. It is available freely now. Only thing is that you have to teach so that your student can understand and use this knowledge which is available. If they don't want to do the research, at least they know how this science has developed and how it is progressed. How can I put it in my practice? Without knowledge, you cannot. Sometimes you will see these details are given with the evidence, high evidence. The level 1A will be there in that column, level 1A. If I'm there, I say, God, this is the knowledge I'm going to believe because it is priority-wise high. That means more believable. They have tons of data and they have said, this is definitely going to work. If I see level 4, then I say, okay, I read it. But science tells me, don't use it. Yet we are not there. So when you see these tables, which I told you, sometimes you are going to see evidence level, high level, moderate level. Then you see here, I have given you the genes, number of drugs, 24 drugs are there. And all the drugs that you will see here, oh God, I have a good chance. Science has progressed. We have solved the problem. See, but when I'm going to come here and all that, not applicable, oh God, I cannot. Yet research has to come. People have to prove, put more data. And then you develop GKB, it will be more and more and more. This is what you see here. This is about the drugs that I told you about the science, psychiatry, neurology, and what percentages are there. I would like to wind up. This is about the IGNITE, what the IGNITE gives you that I've talked about. And this will be my last slide. I don't want to overboard, but I told you, United States government before 12 years, I attended one conference. The lead was given by a cardiologist who was the chief, one person. He has to employ the things. I attended his 6 lectures. Every month he used to come to Chicago. I was there and I started. So I followed since then. And then these are the new. So where these centers are there, what are their names, what they do. That is what India has to do. Indian map, where the scientists are there. This group, this university, AMRAVATI, you focus on this. You go Mumbai, you focus on this. You go Bangalore, you focus on this. We have to have a planning and all. Then all the people have to move together so that one day we cannot say that we make so many drugs, but how to use them, we don't know. It's a race. India has to take that. We have to generate. We have a lot of good minds. We are genius. Focused aim, not money, focused aim. Take the opportunity. I want to develop this. If we do that, we will be number one. It is possible. Only thing is planning how to stick to that and the mass that we have to generate. When I was studying, then people used to say, India is behind 100 years. See, anything in the US, after 100 years, it will come there. When I was doing my BS in 1968, I studied DNA structure and I was thinking, my goodness, what is this? Now DNA structure, any donkey knows. Science has moved so far. See, so it looks like that. Oh God, America is so much advanced. No, we can catch them. If you have decision in 5 years, I will show you the map of India in my another international conference. I said, this is where the India is there. But somebody has to take lead. Somebody has to make it possible. Then India will move fast because we have brains and brains and brains. I'm an Indian guy. I'm not American. See, what is the advantage? I have this opportunity. Make this opportunity available in India. You are like me. So I will just take it out. And I have tons of slides. I wanted to teach you about all application part so that you can understand how the knowledge was taken it by CPIC and what they have. So you have to bear with me because of the time. I have to wind it up and I will declare that if you have any questions, I'll take your questions, please.
Thank you, sir, for giving this wonderful session. Pharmacogenomics in relation to the basic, fundamental, and research applied knowledge you have shared with all our participants. I'm very much thankful on the behalf of the Dr. Rajendra GODE Institute of Pharmacy and Dr. Rajendra GODE College of Pharmacy. Now, I would like to request Professor GULHANE sir to please conduct the session of question answer. Before that, I would like to request Dr. BALWANT CHAUHAN sir to please switch off their slide sharing so that the audience are also visible to them also.
Okay, so before I share, I would like to thank you. Okay.
Thank you, sir.
I hope that I have not wasted your time.
No,
and
no.
for your patience,
This
sir.
was
Thank
a
you.
very wonderful session, sir.
Okay. So now you want me, I have come out. Is it okay now?
Yes, sir.
Okay.
All are visible now, right now. There
Mhm.
one question on our YouTube channel was asked by Dr. Surendra Agrawal, sir, that is related to the DNA microarray technology. He was asking that how to differentiate the colors which are not plotted as per the command. That means the problematic genes and the alleles, whatever detected. Apart from that, if the colors are appeared in a microarray technology, how to detect it?
Well, you saw that slide, and then I showed you few colors. And if you see the spectrum, there were points or dots where there were no color. Am I correct? So
Yeah.
where there are no colors as such, the genes has not been detected.
Okay.
I don't know whether you asked me the same question or you can repeat the question. I gave you, do you remember that you have a normal color, which is a green color, and then you have mutated, then you have a red color. And if you have in between, let's say you have two copies, then it will give you yellow. That yellow color is selected. These are dye. This dye gives you the color. So when you mix what we call it green color and the red color, if you mix it, it will be like yellow. So you have some copy which is yet normal, some copy is mutated. So yellow is in between.
Okay.
And all these color codes which are computerized, there's a program. So when you go and what you need, you have to ask that question and the data will be taken and it will be arranged. Let's say I have the same data which I did it, okay? I have a cell which is normal. I took a cell which is cancer and you ask the question, which genes in these cancers are different than the normal ones? Am I correct? So my red will say that, okay, this is the gene which is found in cancer, which is not found in the normal cell. Normal cell will have a green color and all that. So my all data will be there. And I told you in the background, if you remember, I said this is a colorful and then it has a lot more value, the point and all that. I cannot just explain that, believe me, in 1 or 2 minutes, okay? But what I'm telling is the data is there. Now, let's say today, Dr. Ravindra says, Dr. Chauhan, you study this with cancer. Can I have your data? And I say, why? Well, I want to study with these genes are somewhere related to the diabetes or not. You're not to run anything else. Your computer, you go and which are the genes that you are interested now in diabetic condition related, you select. It's a software, you select that. Whatever your data is there, it will arrange. And now instead of cancer, it will say, these are the genes related to your diabetes. So once you do this analysis and all that, and you have 5 people in your institute, the same data, you can generate 10 research paper and 10 people can use it. It's all computerized. Whatever is given there, it will arrange and it will project and give you the statistics. Make sense to you?
Yeah, yeah. Thank you, sir.
Okay.
I think so. Dr. Surendra Agrawal sir get their answer. If there is any query regarding with this, we will connect with Dr. Surendra Agrawal sir for further any type of the query. Now, I would like to hand over the session to the Professor Chaitanya Gullhane sir. If there is any participant would like to interact with our eminent speaker, Dr. Balwant Chauhan sir, they can raise their hand. There is one option is given on your Zoom meeting. There is more. You can press the button of that particular raise hand so that we can get identified you and we will connect with our eminent speaker. Okay, over to you, Professor Gullhane sir.
Thank you, sir. Thank you, Chauhan, sir, for sharing.
Thank you so much.
Thank you, sir, for sharing this emerging knowledge of the field of pharmacogenomics to our budding pharmacist researchers. It will be definitely useful to them. Okay. Now it is the time of question answer session. The participants who is having any query can put their query in the chat box so that I can put it to our respected speaker. Okay, so you can put it in your chat box and you can also interact with our respected guest. Okay. So, sir, first question is from Vedika. How genetics play a role in the field of antibiotic resistance? How genetics play a role in antibiotic resistance? From Vedika.
Well, this is antibiotic genetics in pharmacology. Probably you will study that, which is the normal part, right? In a what we call it resistance to a particular drug and all that kind of a stuff. But whether you have a condition where you are treating it with the same antibiotics, same kind of the bacteria and all that, and your outcome is not there, that's where you think about pharmacogenomics. Each and every scenario that you see, you are not going to apply that. So when you say antibiotics and then you are talking about, if I'm correct, the resistance or why it's not working, in your pharmacology, you might have seen certain what we call receptors are there. There are influx transporters are there, efflux transporters are there. They are mutated. All that you study is a normal science. Am I correct? Some drug will not enter or it will enter and then it will leave the cell because there is a particular structure in the cell membrane and immediately efflux will be there. The drug will be pushed out. So that is all part that you study, which is related to genetics, but as a normal base to understand the mechanism, mode of action of the resistance or the drug action, which is with all, not only antibiotics. I'm talking about several cancer drugs and all that kind where the resistance is there. But the topic that I'm talking about is that you have the same drug which is given, let's say category of antibiotics, and then the same kind of bacteria, and then it's not working. That's where you think about pharmacogenomics. That's where you say, let me have the pattern, the genome study, and see whether somebody has related this drug and a biological marker. And if the biological marker is related, now you have analysis. If you don't, then you have to take that person's cells, extract DNA, send it to the laboratory, and the laboratory will give you the analysis. Once the analysis is there, that question that you ask, various labs will ask you, sir, you are sending this sample. What is your interest? So in that particular form, when you are writing, you can say this particular bacteria resistant, you give guidelines. And that company, if they have this analysis done, when they give you a report, which is like a lab report, it will tell you all this data. That's where antibiotics, the resistance and individual variation that you consider. Otherwise, if it is not individual variation, your pharmacology teacher might have given you a greater detail. This is the drug resistant developed. This is the mode of resistance. There are 5 or 6 different kind of the what we call it, things are related that gives the resistance. So that is a basic pharmacology analysis. Have I answered your question? If not, help me out, ask something more.
Thank you, sir. Hope so. She must get clear with her point. Another question is there from Rajesh or Nagarajan. Due to genetic variation, it is possible that vaccines are not working for all the persons in the same way as they expected, especially in the case of COVID-19. Am I repeat the question,
Yes.
sir?
Yes. Yes.
Due to
Good
genetic
question.
Yes.
Yes. You said COVID-19. Am I correct?
Yes, sir. Yes.
Okay.
Right.
So have you followed that as the things came? The most important thing that came in the news was respiratory ARDS, pulmonary pneumonia kind of a disease. Am I correct? Many people died because of that. Yes?
Yes, sir.
Hello?
Yes, sir. Yes, sir.
Right? So the news where the people are dying, and that's why they say, do you have a respirator? This country doesn't have a respirator. Why you need a respirator? It's a pulmonary disorder, corona and pulmonary disorder. ACE2 inhibitor in your pneumocyte 2, and then it has a receptor.
Yes,
but later, now they are reporting this person has bleeding problem because of corona-19. This person has a heart problem because of corona. This person is vomiting. Now you read, and now you are going to see different outcomes, not only related to lung. That's where the pharmacogenomics. So you will say this guy, when corona goes, it affects the lung. Nothing happened to the blood system, nothing happened to the heart, nothing happened to the brain. Then you analyze that person and you will find a gene which will be probably responding to the brain. Brain cells have ACE2 inhibitor too. So it goes and binds. Kidney has ACE2 inhibitor. So theoretically, coronavirus should also affect kidney. How many cases you studied people died with the kidney? Are you getting? How many people died with a heart problem?
Yes.
But then you will say most of the people, coronavirus, as if it binds in the lung and nowhere else. No, that's not the case. It binds in the kidney, it binds in the liver, it binds in pancreas, it binds in GI tract. Because it binds in the GI tract, you might have studied that several people develop diarrhea.
Yes, right.
That's where the pharmacogenomic comes. So now you have coronavirus, but your patient has a very specific, the heart is not involved, brain is not involved, lung is not involved. Only the GI tract problem is there. Okay, get this. And now people are working. Let me sequence. What is the difference between that ACE2 inhibitor in intestine? There has to be something different. Most of the virus binds there and gives you that symptom. Few will bind to the lung, few will bind to the heart, few will bind to the kidney, few will bind to the, but effect is not there. So now science is studying. Many people have seen several grants in California. There is a whole bunch of the what we call it grant. They invite only on corona and stem cells, and there is no limit. Millions, 2 million you will get as a grant. And if I see this report, that's where they say why this person corona went to GI tract, not to the lung, how that is happening. Then you compare these people, and that is the future. They will compare what happened in the lung, they will compare what happened in the intestine, and they will find out, oh, this person has genes, and if you have these genes, you will develop diarrhea. You will have no problem with this. You will have no problem with heart. But currently, if you ask me, have I seen? I haven't seen that. People are working. Answer is yes. Any question?
Yes, sir. Thank you for the answer. Vedika, if you're having any question, you can directly interact with our respected sir by unmuting your mic. So her question is again that she's asking regarding the accuracy of pharmacogenetic testing.
Okay. Do you remember I gave you the last slide, accuracy, the categories are given, right? Last slide I said, okay, preliminary. That means yet the data is not there, science is not, we know, we have suspicion, but that GKB side will say, don't take this particular point and start treating your patient. So there were 4 categories. If you remember, it was a vertical collar. And on the top I showed you, it will tell you that. And that means they have studied complete detail and the science says, yes, this is the way you will treat your patient. So that tells you how confident we are in that science of pharmacogenomics. It's a growing science. So in every category of drugs, you go to this GKB, which I told you, it will list that level, high level, medium level, low level, and all that. Higher the level, you have more confidence. So you ask me how confident we are or confidence level. Yes, each and every one you cannot apply the same way. You have to know the science and then put that this science we have enough knowledge and we have too much trust. If you treat it, this is the outcome. You believe that treatment and start treating that. And in some hospitals in United States, they have made it mandatory. You have to consider and you have to treat that because science has given that positive result. Where it is in between or low, that hospital or body will say, no, we know, let the time pass, let we have more data and see the level. Whether it reaches to high level, then my hospital will follow that. Otherwise, I'll not take that list. So it is 1-to-1. If your patient is dying, no options are there, then doctor is going to say, this is the last way I can. Though the hospital is not there, then you go and take the forms and all that. Responsibility of that patient. As a doctor, I said, I'm not sure, but this is what it says. Would you like to try? In another case, I'm going to say, this is what I know. This is what I'm going to try. I'm really confident it is going to work. And they are called in GKB, every site that I gave you, CEPIC is a different term. For GKB, they are called levels. Higher the level, more confident you are in this science. Any other question?
Thank you, sir. If anybody is having the question, please put it in the chat box so that I can convey it to the sir.
Okay, thank you, Gulhane sir. If there is a question, get may ask during this afterward session. We are having one more option for that. There is a daily session feedback is comments over here. The link will be put in a chat box by Professor Yadav sir right now afterward. So that if there is any question asked by the audience, it will be conveyed to the sir and sir will respond to them afterward. Okay, so
Yeah.
it's the time now. Yes, sir.
If I can add, if you permit me to do so, all the participants, I think your conference is for 3 days. I have given my email. So you have that email. So it's not that you have to ask me the question today. You are free to ask the question to me, but then you send me your question via email. Okay? And through your email, if I have an answer, I'll type it in and then I'll give you the reply that this is my answer. So you can do before the conference ends. Any day, any question is coming, go to your organizer, whoever it is, and say, I have to have a Dr. Chauhan's email. You take that email I gave you, and then you communicate via email. And once I have your question, and if I have answered, I'll write down.
Okay, thank you, sir. It is your kind guidance for us and giving us such kind of the option to get access to our participant directly with you. Now it's a time for the valedictory function of today's session. So right now I would like to invite Professor Rushali Panchal Madam to unmute her mic and conduct the session of valedictory function.
You want me to be online or?
Yeah, sir, for just for 2 minutes.
Okay.
Yes, sir.
No, I can be here for 10 minutes, but just let me know. Okay.
Good morning to all and good evening to our respected guest. I am
Thank you.
Professor Rushali Panchale from Dr. Rajendra GODE Institute of Pharmacy, AMRAVATI. So as we come to the end of today's webinar, I take this opportunity to express a virtue of gratitude to our honorable speaker, Dr. BALWANT CHAUHAN sir, for graciously accepting our invitation to share his immense knowledge with such a ease and delicacy. You made us aware of the need and importance of pharmacogenomics in education for healthcare providers and health professionals, including pharmacy. We are really thankful to you, sir, as the session was super interactive, super informative, and super energetic, which updated our knowledge globally about the topic. It was a privilege to listen to you, sir.
Thank you. Thank you.
I now extend my heartiest thanks to our beloved President, Honorable Yogendraji GODE sir, who always add his gesture of encouragement whenever we organize such kind of events. I am also thankful to Dr. R.L. BAKAL sir, Principal Dr. Rajendra GODE Institute of Pharmacy, and Professor H.S. SAWARKAR sir, Principal Dr. Rajendra GODE College of Pharmacy, who acted as a source of motivation and support to us. I now thank to our organizing committee members, Professor PRASHANT AJMIRE sir, Professor SAGAR ANDE sir, and Professor CHAITANYA GULHANE sir, who took an initiative to arrange this webinar series and took all efforts to make today's webinar successful, and also my other staff members who contributed to it. And last but not the least, I thank to all the participants, faculties, research fellows, students, everyone related to this webinar series who has shown their response to this global webinar. Thanks for showing interest, and I hope it will add to your knowledge and be beneficial to you in future. Your responses will keep us motivating throughout the webinar series. Thanks to all. Now I hereby declare the end of today's webinar. We will tomorrow witness the second webinar in the series. So see you then. Stay home, stay safe, stay healthy. Thank you. Yeah, yes, sir.
To young people sitting in the audience, as an Indian, this is what I have to say. Believe in yourself, remain focused.
Thank you.
We have to bring our nation to that level. So my son will say, I want to go back to India, or my relative will say, I'm not going USA. Let us do that. Let us do that.
Yes,
Okay?
sir, definitely.
Yes.
Yeah.
Thank you.
Thank you for today's session, sir, for giving your valuable time. We will be in touch with you for in future also to get your support and guidance from you regarding with the pharmacogenomics. Thank
Anytime.
you.
Anytime, if you need me, you have the details. Wherever I can help, and I believe friendship is not for one moment. Friendship never ends. That's what I believe. So in future, any student has my email. Feel free. I'm not expert in everything. Whatever the way I can help, I will help. If nothing, my Indian spirit will help you. You come to this town, you have nobody, write to me. I'll pick you up from the airport.
Yeah.
That's my word.
Thank you, sir, for your kindness. This is wonderful talk during this session. Right now, this is the time for concluding today's session. Feedback form will get available in a chat box, WhatsApp, and YouTube channels. So you have to fill it. It will be accounted for the final distribution of the certificate for this webinar series. So see you later. Join us on tomorrow's session. After before the 8 hours of today's session, you will get the link for the tomorrow's session. Okay. Thank you, sir. Thank you very much for supporting and stay connected with us and inspiring Indians to become a good researcher and able to stand in front of the developed countries.
Yes, good human, good Bharatiya. We are Indian first. That's our spirit. Young people have to stick to that. So my teaching pharmacogenomics is one part. My second part is take the challenge, lead the world. We have capability and we will do it if we are determined as an Indian. That's my last word. Thank you all. I'll leave the meeting now.
Okay, good night, sir.
Good night.
Stay safe.